So this chapter is divided into 3 parts basically. First is introduction of carbohydrates, and this includes you have to learn about how you draw the structures of most of the carbohydrates, number of carbons present in carbohydrates. You have to learn about aldoses and ketoses. So I'll go through the slides once for a minute, and then we can start from slide 1. Okay, so this is how we define the carbohydrate, having a general formula CnH2O, and then aldoses and ketoses. They are two different forms of carbohydrate, one having the aldehyde group and one having a ketose group, which is ketone. I talked a little bit about enantiomers when I talked about amino acids, that they have superimposable or non-superimposable mirror images, and same comes with the diastereomers and drawing monosaccharides. Okay, this will be most of the learning part. You have to learn how to draw the structures of monosaccharides, and once we learn monosaccharides, we have to learn about the number of carbons, what are epimers, what are anomers, and different structures of monosaccharides. They have different carbon in them, and then we learn about hemiacetals and hemiketals, which are two different forms. Mostly we learn about glucose and fructose, which are monosaccharides, and then disaccharide. Disaccharide will include different forms like sucrose, maltose, galactose, trehalose, and other sugars, and then polysaccharide, which include homopolysaccharide and heteropolysaccharides. Okay, once we are done with that, we'll learn about some modified polysaccharides like chitin, N-acetylglucosamine, glycosaminoglycans, and so on, and some of the examples of them. Okay, and then finally end of the class. Okay, so let's start with the first thing, which is introduction of carbohydrates. So carbohydrates are named so because of they are hydrates of carbon. Hydrates means water. Okay, hydrate or hydro um, hydrates are called like carbohydrates are hydrates of CO2. Okay, so they will have a CO2 group, which is carbon dioxide, and water, which is H2O. Okay, they have a general formula, which is CnH2n, CnH2ON. Okay, and the way you say it is like this. I'll try to write everything on my iPad so that you can make notes and you have the slides on your side. Okay, so imagine if you write general formula of carbohydrates, which is CnH2ON. Okay, this is the general formula of carbohydrates. So if I give you n equals to 2, can you tell me what is the structural formula of the carbohydrate we'll get? Yes, so if I write n is equal to 2, which is C3, or okay, C2H2O whole twice, right? This will be your general formula, and this equals C2H4O2, right? So this is second 2 carbon monosaccharide carbohydrate. Okay, if I write n equal to 3, can you tell me the formula, final formula here? It will be C3H, yes, C3H2O whole 3, C3H6O3, n equal to 4, C4H2O whole 4, which is C4H8O4. Okay, this is how you can say all of them are carbohydrates, because they have a general formula of CnH2ON. Okay, it has equal number of water molecules and equal number of CO2 molecules in it. Okay, and that's how you write general formula of any carbohydrate. Okay, it could be as small as 9 grams per mole. This is the molecular weight. Okay, whenever you see anything in gram per mole, this is basically molecular weight, which is represented by mw. Okay, so it could be as small as 90, or it could be as long as 200 1000 okay, grams per mole. Okay, this should be a large carbohydrate and this would be a small carbohydrate. Okay, so depending upon the molecular weight, carbohydrate could be as small as 90 grams per mole or as long as 200,000 grams per mole. Okay, the second thing is, what are the applications or how we store carbohydrate? What is the function of carbohydrate? Carbohydrates are mostly energy source. Okay, we also store carbohydrate in our body in the form of energy source. Okay, so whenever we have scarcity of energy, we will use those carbohydrates in order to generate energy in the form of ATP. Okay, and the stored carbohydrate in human beings are, anyone? We store carbohydrate in the form of glycogen. Excellent answer. Sugar, exactly. So glycogen is a form of sugar, which is glucose. Okay, so we store glycogen in our body, and there are two different organs where we store glycogen. Anyone? Muscles and liver. Exactly, liver and muscles. They are two different organs where we store our carbohydrate. Okay, and fat, it can be stored all over the body, but carbohydrate are mostly stored in the form of glycogen in our liver and our muscles. Okay, so whenever we require energy, it will be released from muscles. No, it will be released from liver first. Once your glycogen from liver is over, that's the instant source of glycogen. It will be start releasing from muscles. Okay, and this is how you have the metabolism going on in body. So whenever you start fasting, carbohydrates are the source which will be used up first. Okay, that represents that we have carbohydrate in our body as a stored form of energy. Okay, after carbohydrate, your body will start consuming, you have two other source of energy, which is protein and fat. Right, the second would be fat. Right, protein would be the last thing that your body will start burning. Okay, because protein is the structural framework of your body. So most of your muscles, your skeleton, and most of the basic structural framework of your body is made up of protein. So that's why body will use that at the last. Okay, when you have all your carbohydrates over, all your fat is over, then it will start using proteins, and when we'll start using proteins, it will form ketone bodies. And when you see ketone bodies in your urine, because something is wrong in your metabolism, your body is using protein for the source of energy. Okay, and that's why we are conscious about when we get ketone bodies in fasting. uh, Another thing is, they are a structural component of cell walls and exoskeletons. Exoskeletons, I'll focus more on that, because have you ever seen an insect? They have a very hard outer covering, which is called exoskeleton. Okay, it is made up of carbohydrate, which is chitin, and they are very strong. So if you see a cockroach and you press your leg over it for some time, release, it will not die immediately. After some time, it will again wiggle and go away. Right, because they have a very hard exoskeleton. Nature made them very strong in the form of their exoskeleton, arthropods. Okay, informational molecules in cell signaling. Okay, so whenever you induce a signal into the cell, they will form number of molecules as secondary messenger or tertiary messenger. Those are mostly the form of carbohydrates. Okay, and we'll discuss more about that if we learn cell signaling in cell cycle. Okay, but anyway, these are different functions of carbohydrate. The second thing is, I think we are up to here. The second thing we'll focus is aldoses and ketoses. Okay, so carbohydrates are present in two forms. Either they are aldehydes or they are ketones. Okay, if you know basic general chemistry, you might be knowing the difference between aldehyde and ketones. Right, aldehyde is basically CHO group. Okay, if you don't know what is aldehyde, aldehyde is basically CHO. Okay, so the simplest aldehyde is HCHO, which is formaldehyde. Okay, and if you go one more carbon, CH3CHO, which is acetaldehyde. Okay, and so on. If you go through ketone, you at least need 3 carbons in order to make a ketone. Why? Because ketone is represented, if you see aldehyde, they are represented as this. Okay, this group is representative as aldehyde, which is CHO. Okay, if you go towards ketones, at least you minimally you require 3 carbons in order to make a ketone. Why? You need carbon in both the sides. This is a keto group. Okay, this group is represented as keto group, which is C double bond O. Okay, this is called keto and this is called aldehyde. Okay, can you see my writings? Yes. So if you ask me about aldehyde and ketones, I'll say this is the basic difference between aldehyde and ketones with a CHO group and CO group with 2 different carbons on the 2 sides. Okay, and that's how we represent aldehyde and ketones here in sugar. Can you see this C double bond O with the H? This is aldehyde, right? And can you see this C double bond O with 1 carbon over here and other carbon over here? This is a keto group. Okay, this is the basic difference between aldehyde and ketones. Okay, so from here now you can easily differentiate between a carbohydrate having a keto group and carbohydrate having a aldehyde group. Okay, any carbohydrate which has a aldehyde group are called aldoses. Okay, and with a keto group they are called ketoses. Okay, so aldehyde and ketones are present in carbohydrates in this manner. Okay, let's see glyceraldehyde, which is the simplest sugar you have. Glycerol, if I ask you a question, name the simplest carbohydrate you know, your answer will be like simplest amino acid was glycine, simplest carbohydrate you will know is glyceraldehyde. Okay, and this is a ketose. It's also called as keto triose because they have 3 carbon, carbon 1, carbon 2 and carbon 3, carbon 1, 2 and 3. Okay, they are all 3 carbon molecules. Okay, so if I ask you another question, what is the minimum number of carbon a carbohydrate can carry is 3. Okay, you need minimum 3 carbons in order to make a carbohydrate. Okay, so that could be another question. The next thing is enantiomers. Okay, I talked about enantiomers when I was teaching amino acids chapter, right? So enantiomers are isomers, but they have non-superimposable mirror images. Okay, I'll tell you about what a non-superimposable mirror image is. If you imagine a mirror between 2 of my hands, you could see a mirror image, right? Can you see if you place a mirror of my this hand, you will see exactly a same hand over here. Okay, and they are mirror images of each other. But are those mirror images superimposable? Superimposable means can you put this one above the other and it looks the same, it overlaps completely? No. So this is non-superimposable mirror images. But can you say a word which is superimposable completely? If you take a mirror image of an alphabet, it's superimposable with each other. So imagine take a letter A, place a mirror over here, you will have a mirror image. Is it superimposable if I place this with another? Yes or no? Okay, I can show you with drawing a simple figure of how a superimposable mirror images looks like. If I place alphabet A and if I place a mirror which represents like this, you will get a mirror image of the same thing like this exactly, right? Are these mirror images superimposable? Yes, if you place this over this, it's exactly the same. It will overlap each other completely, right? Like this. So these are superimposable mirror images. These are superimposable mirror images. Okay, but if they are non superimposable, they are called enantiomers. Okay, but these are for the molecules. Can you tell me another alphabet which is superimposable completely? I is superimposable. Yes. Another? O? Yes. Is B superimposable? No, because B will have a reflection like this, okay, and these two are not the same, right? Any other image which you think is superimposable? Any alphabet? T is superimposable. Anything else? V? Yes, sir. V is also superimposable. Okay. U is also superimposable. W is superimposable. Z? No. A. Yeah, we started with A. Good. So these are superimposable mirror images. Okay. So let's understand what is a non-superimposable mirror images, what we call as enantiomers. Okay. So can you see this image? This is the structure of glyceraldehyde. If you put a mirror, this will go here, like uh, uh, in the front, and these are the aldehyde groups. This is hydrogen, and this is CH2OH. So is this image superimposable with the other image? No, because they have 4 different functional groups and they do not look same in the mirror, and that's why they are non superimposable mirror images. Okay, and whenever you see two different molecule with non superimposable mirror images, you call them enantiomers. Okay, that's the definition of enantiomers. So stereoisomers that are non superimposable mirror images are called enantiomers. Okay, cool. So we have two different forms of enantiomers. One is called D and another is called L. Okay, and this D and L are based upon the position of OH group. Okay, so imagine if you have a OH group on the right side, we call it a D. Okay, if it's in the left side, we call is a L. Okay, and there are two different forms of enantiomers, L and D forms. L and D isomers are enantiomers. Okay, I'll explain you more about D and L form when we practice more structures. Okay, more and more enantiomers. Okay, let's understand with another simple structure, which is a monosaccharide, and I'll ask you which form is D and which form is L. Can you see the functional group, which is hydroxyl group? Right, this is present on the right side or left side? Right. Okay, and this is called a D form. Okay, and this is present on the left side, and you can call it as L form. Okay, D and L. If you see, L form and D form can be explained in different ways, but this is how I remember, and it's correct most of the time. Okay, so these are D and L forms. Can you explain with another structure? This is again on the right side, and this is D, and this is left side, which is L. This is L glyceraldehyde and D glyceraldehyde. Okay, similarly we will learn how to name glucose as D glucose, L glucose, D threose, L threose, D ribose, L ribose. Okay, these are different names of the monosaccharides we will learn. Okay, I'll go ahead and start with diastereomers. Diastereomers are not mirror images of each other. Okay, diastereomers are not mirror images of each other. Okay, so not every image can have a mirror image that looks the same. Okay, imagine if you have 4 different functional groups, but they are attached with the carbon in a different way, they could be non-mirror images of each other. Okay, if they are not mirror images, they are called diastereomers. Okay, if they are mirror images but they are not non-superimposable, they are called as enantiomers. Okay, and this is the basic difference between enantiomers and diastereomers. Okay, so let's understand the other thing. There are examples of D-aldoloses, which is having 3 carbons and 4 carbons. Okay, can you see your OH? It's on the right side, that's why they are D. Can you see the OH which is on the right side? They are D. Okay, and there could be a question here. There is a OH which is also in the left side, right? Why they are not L forms? So the carbon which you look up to is farthest from the carboxyl group. Okay, so your carboxyl group is the first one, and you see the farthest, which is 1 2 3, third carbon. Okay, and you will look at the position of OH at the farthest carbon from the carboxyl group, which is 3 in this case. Okay, you will learn how we can name these if we take more number of carbons. Okay, so let's move ahead and see this structure, which is epimers of glucose. Can you identify the carbon where we look up to the hydroxyl groups? This is the carboxyl group, and the farthest carbon is here, and this is your OH group. So this is D form of mannose, which is another monosaccharide. Let's start from glucose. Can you identify the farthest carbon, which is here? It's on the right side, which is a D. Another CHO farthest is here, another a D. Okay, if you move the positions, you will find L and D forms. Okay, we are clear up to here? There is a new term introduced which is called epimers. Okay, I definitely remember this by just saying one carbon difference. Okay, I remember epimers as one carbon difference. What is that one carbon difference? You have to take this glucose molecule as your reference. Okay, and you have to learn this molecule, how to draw the structure of glucose. Which is one of the most common molecule we will learn in carbohydrates. Okay, glucose, I say 3 at third position, OH in the left side, other one OH is the right side. Okay, this is how I remember. So if you see structure of glucose, can you see third carbon OH is on the left side and others are on the right side? Okay, can you remember that? Third carbon OH is left and all others are on the right side. Okay, so now you take this as a reference and compare with the other two molecule, which is D mannose and D galactose. Okay, can you see a difference? 3 carbon OH is on the left side, no difference. 4 carbon, 5th carbon, 6th carbon, everything is fine. Can you see a difference? Here we have a position difference of hydroxyl group in the second carbon. Okay, that's why they are called epimers at C2. And I said epimers are one carbon difference, which means the position of hydroxyl group will be different at one carbon. Okay, and that's why they are called epimers. Am I clear up to here? Epimers are the same structural forms of glucose or any other molecule having one carbon hydroxyl group difference. Okay, so the hydroxyl group at second carbon is on the left side, then that's why they are called as epimers of glucose. Okay, and this is 2 carbon epimers. Can you see the other molecule, which is galactose? Can you take this as a reference? OH right side, H right side, OH, this is changed, right? Which position? 4th carbon. Okay, and this one is 2 carbon. Okay, and this is how you have to remember. I remember this as a very simple, because I don't want to remember this now and forget it later. I remember 4G. Okay, 4G is the fastest network. Okay, if you have difference at carbon 4 is galactose. Okay, and I remember 2M. Okay, 4G and 2M. I just remember 2 minutes mostly, 2 minutes and 4G. Okay, 2 minutes, 2 carbon difference is mannose, 4 carbon difference is galactose. But the first thing you have to remember is structure of glucose. Okay, I'll go one more time to help you draw the structure of glucose. Okay, so if you don't know anything, just start from the scratch. Okay, we know glucose is a 6 carbon molecule, right? So draw 6 carbon, 1 2 3 4 5 and 6. Okay, I don't know anything, I'll just draw the 6 carbon chain and we'll start the numbering, which is 1 2 3 4 5 6. Okay, as we know, we are drawing the structure of glucose, which is a aldose. Okay, which is a aldose, which means it has a CHO group, which is aldehyde. So I'll draw the first carbon as CHO. Okay, and the last carbon will be always a CH2OH. That's how we draw the structure of any carbohydrate, right? And then now we have to draw this. We have to put the structures of H and OH on the other sides. Okay, that's what it is left. So we'll draw two lines from each carbon, and now we have to decide where we have to put a H and where we have to put a OH. Okay, so I told you if you want to remember the structure of glucose, you have to remember the third carbon. The difference is only in the third carbon, and you will see 1 2 3 3 OH is left. Okay, 3 OH is left, and other OH are on the right side. Okay, OH, OH, OH. Okay, and whatever is left is hydrogen. Okay, which is H, 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 and H. Okay, is it comfortable now? You can draw the structure of glucose. The only thing you have to remember is third carbon, OH on the left side. Okay, and other OH is on the right side. That's it. Okay, now I'm not satisfied whether I draw the structures correct or not. So the only thing I have to remember is structure of molecular formula of glucose, which is C6. I need answer. C6, H12, and O6. That's the formula of glucose, right? Can you count the number of carbons? 6, 1 2 3 4 5 6. Can you count the number of oxygen? It's 1 2 3 4 5 and 6, right? C6, H6. And can you count the number of hydrogen we have? 1 2 3 4 5 6 7 8 9 10 11 and 12. Okay, so our structure is correct, right? C6, H12, and O6. Now I'll ask you to draw the structure of mannose. Okay, I said 2 minutes. It will take 2 minutes to draw the structure of mannose, right? And 2 minutes is 2 is carbon. So the only difference is at second carbon. So second carbon is here, right? Second carbon is here. What's the difference? OH is on the left side. So let me erase this one, and let me put OH over here and H over here, and this is mannose. This is L mannose or D mannose? D mannose. Why D mannose? Because at the fifth carbon, hydroxyl group is on the right side. Okay, cool. Am I clear up to here? Can I ask you to draw the structure of galactose? I told you to remember with 4G, and the only difference should be here at 3 carbon. No, the difference would be at 4 carbon, right, which is here. So I'll remove OH and I'll put a OH over here, and that's it. Okay, but you have forgot to change the 2 carbon, right? It should be at the same position, which is OH over here and H over here, and this is now not mannose but galactose, right? Cool. So let's move ahead and see the other structures, how we can remember. So now you know the about epimers, right? If I ask you a question, mannose, tell me whether this statement is correct or incorrect. Okay, mannose is 4 carbon epimer of glucose. It's incorrect. Mannose is 2 carbon epimer of glucose. Correct. Galactose is 2 carbon epimer of glucose. Incorrect. Mannose is 4 carbon epimer of glucose. Correct, right? Now you can remember the names of the epimers we have, right? And next, after some time, we'll see another term which is anomers. Okay, epimers is 1 carbon difference. I'll tell you what is the difference between anomers. Okay, which is 2 different forms of glucose. But let's move ahead. Okay, structures we have to know is ribose, glucose, galactose. Galactose is epimer of glucose. Mannose is also epimer of glucose. Fructose and ketose are form of glucose. We'll learn this one and one. Okay, ribose and other sugars too. But let's move ahead and see different aldoses we have. Aldoses are carbohydrates having a aldehyde group. Okay, carbohydrates have a aldehyde group are called aldoses. I said simplest aldose or sugar could be a 3 carbon sugar. Okay, if you see a 2 carbon sugar, it's not possible, right? If I say minimum carbon can be 2 in a carbohydrate, it's an incorrect statement, right? Minimum carbon could be 3. So carbohydrate starts with 3 carbon molecule. So this is 1, 2 and 3, and this is a D form. Why? Because OH on the right side. Let's see another D3Os, which is D because the last carbon has OH on the right side, and it's 1, 2, 3 and 4 carbon sugar, right? 1, 2, 3, 4 carbon sugar. Okay, erythriose and threose. Okay, they are cis and trans form of the same sugar. Can you see that? Both OH are present on the same side, so it's basically a cis form, and OH and OH are present on the other side, which is a trans form. Okay, this is cis and this is trans. Okay, and you can easily remember that. Why? Can you write letter E? See, if you consider these two groups on the same side, this is letter E. Okay, which is cis form, which is erythriose. And if you write letter T, both can be on the opposite side, which is threose. Okay, so trans is threose, cis is erythriose. Okay, can you remember that those two structures? Yes. Let's move ahead and see aldoses with 5 carbons. Okay, we have learned 3 carbon, which is here, 3 carbon, we have learned 4 carbons, and now 5 carbon. Okay, and 5 carbon, the most important you have to remember is D ribose. Okay, why D ribose is important? Because you will see that in a lot of structures, which is RUBP, ribulose bisphosphate. Okay, in citric acid cycle or photosynthesis, you will see these molecules. I don't remember, you need to understand those things, but yeah, ribose is an important sugar. Okay, and you just have to remember how OH groups are attached to the carbons. Okay, and that's the only difference between all those forms of sugar. Okay, so imagine how nature created those molecules having different properties with different functional groups attached on the other sides. Okay, can you see that? They have different properties, they are named differently, but they have the same structural formula, just different arrangement of hydroxyl group from one carbon to the other, right? So if you see the next structure, which is ribose, all hydroxyl group are on the same side. This one is at different side. This is one is different side, but in the middle. This is two on the different side, and this is the same. But you see a common thing, which is OH is always on the right side. That's why they are all D forms. Can you see that? They are all D forms of sugar. Okay, so you just have to remember it in the some or the other way, but I don't think you have to remember each and every structure correctly. I don't think that it will be asked in your exams, but still you are curious to know, then you can remember them. Okay, but D ribose, all the OH on the right side. Okay, similarly, aldoses with 6 carbons are here, and the most important are glucose. We already learned the structure of glucose. Mannose we did learn, galactose we did learn. Okay, if you are more interested, you can see the structure of gulose, idose, talose, ultrose or allose. Okay, allose is all on the same side. This one different side, this one second different side, and this one third different side. This one is second and fourth, this one is second and third, this one is all three. Okay, but if you see, all the forms are D forms. Why? If you focus on the last carbon, all OH are on the right side, and that's why they are D forms. Okay, am I clear up to here? If you want to remember the structures, you can practice them at home like twice or thrice, and you will be able to remember that, but not necessary for the exams. So we are here up to, we finished up to like aldoses, different types of aldoses, we finished all of them, and now we will move to ketoses. Okay, are my slides and writing visible to everyone? Okay, cool. So we'll now move to ketoses, which are ketone forms of carbohydrates, right? These are the second category of carbohydrates you will learn. So ketosis means they have a ketone group, okay. And if I already explained what is a ketone group, which is C double bond O, and there will be two carbon on the two other sides, okay. If it's one carbon and one hydrogen, it's aldehyde, okay. So whenever you write a structure of ketose, it must have minimum 3 carbons required, okay. Let's understand the first simplest ketoses as carbohydrates are dihydroxyacetone, okay, which is called DHA, dihydroxyacetone, okay. Dihydroxyacetone is as simple as glyceraldehyde. You remember glyceraldehyde with 3 carbon, and this is also 1, 2 and 3 carbon. So if I ask you a question, 3 carbon simplest ketoses as carbohydrates, the name will be dihydroxyacetone, okay. 3 carbon simplest aldehyde as sugar is glyceraldehyde, okay. This is how you remember those names. Let's move ahead to another ketoses with 5 carbon and 6 carbon. The most common and most important is fructose as 6 carbon, okay. 5 carbon ribulose and xylulose, which are present mostly in plants, and 6 carbon sugar are sucrose, sorbose, tagatose and fructose. The most important out of them is fructose, okay. And if you see, fructose and glucose are very similar. Fructose and glucose are very similar. Why? Let's understand about the number of carbons: 1, 2, 3, 4, 5, 6. Yes, this is another 6 carbon molecule, right? And if I remember the structure of glucose, they have CHO in place of CH2OH, right? This whole thing is CHO, and they have CH2OH at the 6th position, okay. I'll draw parallelly the structure of glucose and fructose because they are isomers of each other, okay. So can you see the third carbon? Third carbon OH is on the left side and others are on the right side, okay. So this is the only difference between glucose and fructose, which is presence of ketone group in place of aldehyde group, okay. So can we call them as functional isomers because they have functional group different, right? So one is having an aldehyde group, which is glucose, and one having a ketone group, which is fructose, right? So so glucose and fructose are functional isomers of each other, okay. So let's understand. If you see where so let's draw a fresh structure of glucose, okay. One more time, I'll draw with you, okay. Please draw a structure of glucose molecule: 3 carbon, 4 carbon, 5 carbon, 6 carbon, okay. In glucose you have CHO and this one is CH2OH. Can you remember where we have a difference at OH group? Third carbon, yes or no? Yes, third carbon is 1, 2, 3. Third carbon OH is on the left side and other OH are on the right side. Rest of them are hydrogen. Yes. So this is structure of glucose, right? Specifically D-glucose. Okay. Let's understand the structure of fructose. I will write CH2OH 1, C, 3, 4, 5 and 6 as CH2OH. Can you see I wrote CH2OH and CH2OH on the top and bottom carbon, okay. And I'll definitely write the carbon number, which is 1, 2, 3, 4, 5, okay. If I told you this is an aldehyde group, right, ALD, okay, and I have to write a keto group over here, which is on the second carbon, that's why it's keto, right. Now we have to see hydroxyl group, which is on the left side at third carbon and others will be on the right side, right, and rest of them are filled by hydrogen. Can you count number of carbon from here? 6. Can you count number of hydrogen? 12. Thank you. If it's not 12, I'm wrong. Like I draw the structure wrong. Okay. And oxygen? 6. Do you remember this is still C6H12O6? Yes, sir. So, and this is D or L? D. So, this is D fructose. Am I right? So, this is D glucose and D fructose. The only difference is one is having aldehyde group and one is having a keto group. Okay. Am I clear up to here? Any questions? No. So, let's move ahead and see the another form, which is hemiacetals and hemiketals. This is very important to understand if your sugar or your disaccharide sugar is reducing or non-reducing. Okay, there are two different terms: reducing sugar and non-reducing sugar. These are the very frequent questions in your exam, even in MCAT and other exams. We will have four options. You have to select one of them, which is reducing or non-reducing. Okay, so you have to understand this reaction between aldehyde and alcohol. Okay, and we call them whether this is a hemiacetal structure or acetal structure. Okay, what is hemiacetal and what is acetal are two different terms. So if you see, let me make this big so that you can understand. I'm not sure I can draw. Yeah, I can. Okay, so you have to learn. You have to know the basic understanding of how organic chemistry reactions takes place, right? How electrophile and nucleophile attack on the carbonyl position. Okay, if you see this RCHO bond, which is aldehyde bond, right, which is aldehyde group. Okay, this is aldehyde and this is alcohol. So your hemiacetal and hemiketal structures can be formed between aldehyde and attack of alcohol. Okay, so do you know that this is a partially polarized, this is a polarized bond, and this is carbon and oxygen. Okay, and they have a electronegativity difference. Okay, and out of carbon and oxygen, which is more electronegative? Out of carbon and oxygen, which is more electronegative? Oxygen. Oxygen is more electronegative, and the most electronegative element of the periodic table is fluorine. Okay, fluorine followed by oxygen, followed by nitrogen, followed by carbon, followed by um, hydrogen or any other element. Okay, but the most important is most electronegative element is fluorine. Second most electronegative is oxygen. Third electronegative is nitrogen. Fourth is carbon. Okay, or chlorine. After that, carbon is chlorine, or almost they have same electronegativity. So this is how I remember phone call. Okay, FON. Okay, and then carbon or chlorine, phone call. But anyway, so this is the electronegativity order difference in the periodic table. But this is partial negative charge and this is partial positive charge due to the electronegativity difference they have, right? Are you clear how this happens? With carbon having oxygen, this is partial negative and this is partial positive. As electrons are cloud, okay, if you have an electron cloud between carbon and oxygen, okay, if you have an electron cloud between carbon and oxygen, oxygen is more electronegative. What you mean by more electronegative is they have higher capability of holding electron cloud. They are more powerful. They are more powerful to hold electron cloud. Okay, so if me and what's your name? Eric. If me and Eric, we are friends, okay, and we have a partnership of $50, okay, and he's very powerful than me. He's in El Paso. He's big boss of El Paso, and he's very powerful. So out of my 50 partnership, he always carry $40, and I always carry $10. So he is more electronegative than me, and he's carrying more, always more electron cloud towards himself, and that's why he is always partial negative charge, because electron has a negative charge overall. Okay, so the more electronegative element here is oxygen, and that's how electron is, oxygen is partially electronegative and carbon is partially positive, okay, always. So it means they always have partial negative charge and partial positive charge. If you see alcohol, they have a R group and a OH group. OH is negative and R is positive, because again R could have a carbon and oxygen is more electronegative. So if you attack them, so what do you think? Negative will attack at positive side? Yes, sir. Negative will always attack to the positive side, and positive will attack to the negative side. Okay, and this is, you have a structure of hemiacetal. Okay, can you see this R from here? It's already over here. C, same position. Can you see this hydrogen is over here? This hydrogen is over here, and you have OR. Okay, OR, which is this oxygen and this R. Okay, and you have another group which is OH, which is OH. Can you draw this structure from this reaction? It's easy. I'll draw one more time with a blank page so that you can understand easily. Okay, it's easy to understand when you have a blank face or blank brand or blank copy. Okay, so if you have RC double bond O with hydrogen, and you have two ROH, which is alcohol. Okay, and if you break this, and this is always, let me change the color so that you can see it easily. Can you see this is partial negative charge and this is partial positive charge? Yes, sir. And this is partial negative and this is partial positive. If you break this bond, this will be positive and this will be negative. Okay, positive will attack at negative and negative will attack at positive. Now this structure will be R C, this double bond will break and this will be OR, this is H and this will be OH. It's fine now. So this structure is called hemiacetal structure. Okay, now we have another chance to attack on OH group. Okay, let's understand from this. Now you have another ROH which you can attack on this position. Okay, and let's understand how. You have O and H. Okay, and in oxygen and hydrogen you always have a chance you can break them. Hydrogen as positive, oxygen as negative. So can you remove hydrogen again? Yes, sir, you can. Hydrogen is positive, oxygen is negative. Here you have negative and positive. Okay, this negative will attack on positive and this positive will attack on negative. Okay, now you have OR and OR. You see that? And this will remove a molecule of HOH, which is nothing but water, right? And this is your acetal structure. This is hemiacetal because it's half acetal. That's why I call they hemiacetal. Am I clear up to here? This is a simple reaction, but you have to understand how we form hemiacetal or acetal structures or ketal or hemiketal structures. Okay, clear? Can you practice one with the second group, which is ketone group? Can you practice in your copy the second reaction with ketone group? Form a hemiketal and ketal structures. Form a hemiketal or ketal structures. If you practice with me, it will be more easy for you to learn. If you do not practice, it will be harder to learn when you are practicing at home. Okay, so go with me so that you can learn it faster. Okay, always. And the most traditional way of learning is always have a copy, a pen, and your learning material in front of you. If you just read, you will never be able to learn. Always have a copy and pen with you so that you can always write whenever you learn. This is how memory systems are created in your brain. Okay, hey, can you write a positive and negative between ketone and alcohol? Yes, so practice with me. So this is partial positive and this is partial negative, and if you break this bond, this will be positive and this will be negative. This negative will attack at the positive side and this positive will attack at the negative side. This is R, this is R, which is common. This will be O and R, which is here, and this OH will go over here, right? See that? Can you attack again with molecule of ROH? This is R positive, OH negative. Okay, this OH bond would be H positive, O negative. This will remove a molecule of water, and this will be again OR, right? So this is acetyl and ketyl. One is having a acetaldehyde, acetone group, aldehyde group, and this is having a ketone group. Okay. So here aldehyde or ketone will act as electrophile, and alcohol oxygen atoms will act as nucleophile. Okay, you represent electrophile as E plus and nucleophile as Nu negative. Okay, this is fundamental organic chemistry. Okay, if you are more in chemistry, you will definitely will be able to relate to these reactions. SN1, SN2, E1, E2. Okay, these are common reactions of organic chemistry. But anyway, let's move ahead. Any questions from here? Yes, no? Can you attack one more time here with ROH? No? Can you attack with ROH one more time? Yes or no? No, because there is nothing you can remove. Okay, there's nothing you can remove. You cannot attack one more ROH. That's why they are non-reducing sugars. If you see a complete reduced structure, it's non-reducing sugar. Okay, if you have already $0, it's very hardly you can spend any money. Okay, you can use credit cards though, but yeah. So they are already reduced up to final point. That's why they are non-reducing sugar. We'll learn this after we'll draw the structures of different acids. Okay, and after that I'll give you when we are supposed to have a break at 12. Okay, I think we are 7 minutes behind 12, 6 or 7. Okay, the next thing is cyclization of monosaccharides. Okay, make sure I am going with you. I'm not going ahead, I'm not going fast, I'm not going slow, I'm going with you. Okay, if you are I'm going fast or I'm skipping something, please interrupt me in between and I can turn over again. Okay, I want to go with you so that you can learn with me. Okay, so let's understand cyclization of monosaccharides. Okay, it's very important and it's very easy. You just have to follow the steps and learn how we draw it. Okay, and you will be definitely able to learn. Okay, so it's called cyclization of monosaccharides. I already introduced the term epimer to you. You remember? And I told you we'll learn about anomers. Okay, so epimers and anomers are two different terms you have to learn in order to see the structures of carbohydrate. Okay, so says pentoses and hexoses readily undergo intramolecular cyclization. Intramolecular means within the molecule. Okay, they form the cyclic structures within the molecule, and that's why it's called intramolecular cyclization. Okay, and they form a new chiral center. Okay, when they go under cyclization, they form a new chiral center, and that carbon is called anomeric carbon. Okay, so whenever during process of cyclization there is a new chiral carbon formation, we call them anomeric carbon. Okay, if you don't know what is a chiral center, I'll repeat it again. Whenever you have a carbon attached with 4 different functional groups, they are called chiral carbons. Okay, if you remember my amino acid class, I talked about amino acids have a chiral centers, and that's why they are optically active, right? If you have a chiral center, that you have a chiral carbon, your compound is optically active. Okay, so whenever you go under the process of cyclization, you generate a new carbon which is called chiral carbon, and they are also called as anomeric carbon. Okay, so it could be in the form of alpha and beta. Alpha is transform and beta is cis form. Okay, and we'll see how we can draw alpha and beta together and understand their uh, importance in the structure. Okay, so I'll start with the molecule which is D-glucose, and if I'm confident, I can say that all of you can draw a structure of glucose, right? I'll repeat it one more time, last time, to understand how you can draw a structure of glucose molecule. Okay, I say glucose is a 6 carbon molecule and it contains aldehyde group. So I'll write 1, 2, 3, 4, 5, 6 carbon. Okay, the only thing you have to remember is third carbon OH is on the left side, which is 1, 2, 3, OH is on the left side, and all other OH are on the right side, and your glucose structure is ready. And you must have aldehyde group at the top and CH2OH group at the bottom. Okay, am I clear up to here? So let's kick this molecule from here and drop this on the other side. You know, so this is the molecule of glucose. Okay, I'll kick it from this side. Okay, and it will drop like this. We'll start preparing for sleeping. Can you see that? Now your whole structure will be like this, and you have a carbon CHO over here, and followed by carbon, 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 and CH2OH will be the cap. So cap of the structure is CH2OH, and you will start from here. Can you relate that structure from this? Okay, I'll start with carbon 1. Okay, can you see carbon 1 is CHO? Yes, sir. Can you focus on carbon 2? Okay, now you have to transform one structure which is in the vertical position to one structure which is on the horizontal position. Okay, so if I see any functional group or any hydroxyl group or hydrogen which is present on the right side will go to the bottom. Okay, and anything which is on the left will go to the top. Okay, because I literally push the molecule on the other side. Okay, anything will be on the right side will go to the bottom, anything on the left side will go to the top, right? So can you see the position of H and OH at the second carbon? OH is on the right side, so it will go to the down position. H in the upper left will go to the up. Third carbon is OH on the left side. Third carbon, can you compare it here? The third carbon OH is up. See, third carbon OH is up, which is here. Fourth carbon OH is down, OH is down. Fifth carbon, fifth, okay, sorry, this is this third, this is fourth, this is fifth carbon. Fifth carbon OH, OH is here, right? This OH will react to this, that's why we kept it here. Okay, and your CH2OH will be on the top position. Okay, can you see these two structures are basically same, but they have different positions of H and OH? Yes or no? Yes. So now we have OH, this OH which will react to this and form a cyclic structure. So carbon 1 and carbon 5, cyclization happens between first carbon and fifth carbon. Okay, so let's understand how this cyclization occurs. I already told you about this, how this reacts. If you have CHO, this is partially negative and this is partially positive. Oxygen is negative, hydrogen is positive. So negative will attack positive and it will form a bond, right? And let's understand this COC. This is the bond they will form. Okay, COC. Okay, this carbon, this oxygen, and this carbon, and you have additional hydrogen over here. Okay, and this hydrogen will go over oxygen, and you have a OH and a H. Okay, can you relate this and follow me? Once you draw the structure which is on the horizontal position, you have a oxygen and a hydrogen, right? This oxygen having a negative charge will attack to the positive carbon, and this hydrogen with a positive charge will attack to the negative of oxygen. Okay, and you will have COC bond with a hydrogen and a OH. Okay, this could be the other way. Maybe you will have oxygen hydroxyl group at the top and hydrogen on the left side, on the downside. Okay, and you will have another structure, and that's why you have glucopyranose with a beta form and glucopyranose with alpha form. Okay, you understand me? These are basically same with a difference in the position of hydroxyl group. Okay, This very easy structure, you just have to practice it one more time. Okay, you want me to practice on the whiteboard so that you can learn it, or this is okay? This is okay. Cool. Now you can understand how we do the reactions. If you have alpha, I always remember is alpha inside, beta outside. Okay, alpha is inside, beta is outside. And what is inside and outside is hydroxyl group. If your hydroxyl group is inside, it's alpha, beta is outside. Okay, alpha in, beta, alpha in, beta. Okay, this is how you remember alpha and beta forms of glucopyranose. Pyranose is a cyclic form of glucose. Okay, it's a 6 carbon ring, that's why they are called pyranose. If it's a 5 carbon ring, it will be furanose, which is structure of fructose. Okay, so I think we are clear up to here. We know the structure of glucopyranose with the alpha form and beta form. Alpha is in and beta is out. Okay, so let's understand the second thing, which is pyranose and furanose. How we say? It's a 6 member ring and this is a 5 member ring. Okay, so 6 membered oxygen containing rings are called pyranoses. Okay, I'm sorry, and this is and this is furanoses. Okay, anomeric carbon are drawn on the right side. Okay, what is anomeric carbon in this structure? Anyone? This is very important. So we formed a new carbon, chiral carbon, you remember? That's why we call they are anomeric carbon. So the new carbon, chiral carbon form is this one, because this is carbon, hydrogen, hydrogen, this is other functional group, this is other functional group. So this is new chiral carbon. Okay, and this new chiral carbon is called anomeric carbon. Okay, now you have new carbon in this story, right? Now you have a new chiral carbon in this story. And if you remember the structure of mannose and galactose, the arrangement of H and OH groups were different, and that's why we called them epimers, right? Now you have H and OH positions are different at anomeric carbon, and that's why we call them anomers. Okay, so alpha D glucopyranose and beta D glucopyranose are anomers of each other. Okay, clear? So you can write the sentence: alpha D glucopyranose and beta D glucopyranose are anomers of each other. Okay, so let's see. I think you can now relate with these structures. You have different carbons here, first, second, third. Third is left. When you have a left, you have this on the top. Okay, and this is pyranose. Why? You have 1, 2, 3, 4, 5, 6 member ring. Okay, this is structure of fructofuranose, which is 1, 2, 3, 4, 5 member ring. Member ring is 1, 1, 2, 3, 4, 5. Okay, this carbon is different. If you are more curious, you can draw this structure from fructose. Anyone who remembers the fructose structure of fructose? This is CH2OH, this is CH2OH, and this is double bond O. This is 1, 2, 3 carbon, OH is on the left side and others are right side, and this is structure of fructose or fructose. Okay, again you have to push this, drop this on the horizontal side, and you will be able to draw this structure. Okay, if you can practice, it's easy. Okay, if you want, I can draw, but it's easy. Okay, let's go to see the structure of fructose. Okay, this is fructose. Yes, sir. So drop this structure like this. You will be able to find CH2OH over here. Okay, and you have oxygen, you have carbon, you have carbon, you have carbon, you have carbon, and this is again CH2OH. Okay, first, second, third, first, second, third OH on the left and other right side. Okay, and that's the structure of fructofuranose. Furan because it's a 5 member ring. Fructose, that's why it's its cyclic structure of fructose. Okay, it's easy, but you have to practice a lot so that you can be able to draw it perfectly. Okay, so these are two different structures of glucose and fructose. I think I'm ahead of time, so let's start after the break about reducing and non-reducing sugars. Okay, I think I'm clear up to here. If you have questions, I'm already here, you can ask me, and we can start the class after the break. Okay, so it's 12:07. Let's meet at 12:17. Okay, it's a 10-minute break. Please come back at 12:17.